Today we will study about the Megagametophyte. Also called female gametophyte. The haploid megaspore mark the beginning of the female gametophytic generation. In angiosperm, the female gametophyte is represented by extremely reduced sac-like structure, the embryo, which remain embedded in the sporophytic tissue. It is totally dependent upon the surrounding sporophytic tissue for its nourishment. Development of embryo cell. It is of polygonum type. Of the four megaspore formed by meiotic division in megaspore mother cell, the megaspore lying toward the chalazal end form the embryo sac. Its developmental stage are as follows. The functional megaspore begins to elongate along the micropylar chalazal axis. In the cytoplasm, several small vacuoles makes their appearance which later fuse to form a single large vacuole. Second, the nucleus of the megaspore divide mitotically, forming two daughter nuclei. The spindle of the dividing nucleus is oriented along the long axis of the cell so that the two nuclei faces the two opposite poles. The nuclear division is not followed by wall formation. However, the two nuclei are pushed toward the opposite pole by an expanding central vacuole. Third, both the nuclei now divide twice, giving rise to 4 nuclei at each pole. The 8 nuclei so formed, however, remain embedded in a common cytoplasm. The embryo sac now elongate considerably and undergoes cellular organization. At the micropylar end of the embryo sac are present 4 nuclei which get organized into 3 plus 1 arrangement in which 3 cells form the egg apparatus and the fourth is left free in the cytoplasm of the central cell to form the upper polar nucleus. The egg apparatus consists of one egg and two synergids. The major portion of the egg apparatus is surrounded by the central cell. Only its micropylar end is attached to the wall of the embryo sac. The three cells are almost of equal length. However, the egg cell extends a little more into the central cell. Seventh, at the chalazal end, 3 nuclei out of the 4 join to form the 3 antipodal cell and the fourth nucleus function as the lower polar nucleus. Later, the lower polar nucleus come to lie alongside the upper polar nucleus. The pattern of embryo development is known as polygonum type and is a common occurrence among the majority of angiosperm. About 81% of angiosperm possess this type of embryo sac development. The structure of mature embryo sac. The mature embryo sac is an ellipsoidal structure with a tapering end and possessing 7 cell, 8 nucleated organization, that is the 3 cell egg apparatus, 3 antipodal cell and a binucleated central cell secondary nucleus. The 7 cells of the female gametophyte are formed within the wall of the parent megaspore. The various constituents of the female gametophyte are as follows. They are the embryo sac wall. The wall of the embryo sac present interesting feature. It is considered to be multi-layered and pectocellulosic in nature. The innermost layer is represented by the wall of the functional megaspore, whereas the outer layer comprises crushed cells of the nucellus. In stipa, the gametophytic wall is made up of 3 parts. The innermost portion is contributed by the gametophyte and the middle portion by the degenerate megaspores and the outer portion by the nucellus cell. Egg apparatus. The egg apparatus is present at the micropylar end of the embryo sac and consists of 2 synergids and 1 egg cell. The synergids are elongated structure lying at the micropylar end of the embryo sac and partly enclosing the egg. The cytoplasm of synergid is rich in different organelles. An interesting feature of the cytoplasm is its polarized nature, that is, the chalazal end is occupied by the vacuole, whereas cytoplasm and a prominent nucleus are present in the micropylar half. Each synergid is associated at its micropylar end with a filiform apparatus. The filiform apparatus is a mass of finger-like projection of the synergid wall consisting of tightly packed cellulosic microfibril enclosed by a non-fibrillar sheath. The lifespan of synergid is of short duration. In embryo sac possessing two synergid, one degenerated before the pollen tube entered the embryo sac, whereas other, known as the persistent synergid, degeneration generate after the pollen tube has discharged its male nucleus into the embryo sac. Synergid haustoria have been observed in the number of plants such as Cotula, Sedum, etc. Function of synergids are highly active metabolically and perform three important functions. These secrete chemotrophically active substance which help in directing the growth of the pollen tube. The degenerating synergid form the site for the discharge of the pollen contained. The filiform apparatus of the synergid help in the absorption and conduction of nutrient from the nucleus into the female gametophyte. The egg cell lies slightly above the synergid enclosed by pectocellulosic wall. In the early stage of development, the egg shows polarization of cellular content. The micropylar end of the egg cell is occupied by a large vacuole, whereas the chalazial end is occupied by the organelles. The mature egg also contains large amount of starch which is utilized during fertilization and early embryogeny. In Nicotiana rustica, the egg contains two RNA positive bodies that are involved in the enzyme synthesis required during early embryogeny. In Plumbago capensis, finger-like projection arise from the micropylar end of the egg. In rare case such as in Cortaderia jubata, the egg develop into haustorium. Thank you.